Good afternoon, Nishika. How are you doing? Good. So, today we'll be beginning our talk with the types of neurons. Then we'll be shifting towards neuroglia. Okay. I'm Rahul Saha and welcome to Wisdom 360. And so, last time we had talked about the different various parts and the functions of the neuron, like the axon hillock, the real axon, the soma, right? Also known as the perikaryon or the cell body. Then coming to the telodendria. Now, today, before we begin further, I would like to introduce you to various types of neurons, right? So, there are various types of neurons known as anaxonic or anaxonic, bipolar, unipolar, multipolar. So, there are various types of neuron based on structure, okay? Because then there are based on function, if you do, so there are different criteria. So, criteria here is, or criterion, I should say, is based on structure because there are various criteria to demarcate the types of neurons. If you think of function, then there are sensory neurons, motor neurons, right? Then if you think of the structure only, then there are anaxonic neuron, then there is bipolar neuron, then there is multipolar neuron, right? And then there is unipolar neuron. Put unipolar here. Then multipolar neuron. Now, first of all, we'll be talking about anaxonic neurons, right? The name anaxonic itself says without the axon. So the neurons which are without the axon, they are known as anaxonic neurons. So I'll show you the image of anaxonic neurons. Anaxonic neuron. So these are the four types. You can see right here the four types of neurons. Just see this image, okay? Think it, you are able to see the image. So this is anaxonic. See here there is no axon, the cell body and the processes which are the dendrites and then the dendritic spines that I told you in the last class. Then there is this bipolar neuron, means it is at the center. You can say the two processes are separated by only the cell body, right? Double axon originating from the same cell body or cyton or perikaryon or soma, whatever you want to call it. There are four synonyms for the cell body present in the neuron. So the bipolar neurons have two processes which are separated by the cell body in simpler terms. Then there is the unipolar one, the third one. In case of the unipolar neuron, what happens is you have a single elongated process. See, the process is a single elongated process and it has got demarcated or divided, got segregated after it has got emanated from this specific cyton or cell body. So unipolar neurons, you can say, have or possess a single elongated process with the cell body located at one side. You can see here the cell body is located at one side, and then this has come out, emanated from the cell body, and then it has got segregated into two sides of the two opposite sides of the cell body. So it's a unipolar neuron. The polarity is only one. It has not gone out from the cell body in two different directions directly. So one little stretch is there, one small process, from there it has got divided. And then you have multipolar neurons. Multipolar neurons are all what we know of, right? Mostly the teachers teach the multipolar neuron in the beginning. The best neuron diagram that you have seen is really of a multipolar neuron because that's the most common type of neuron. Okay. So in multipolar neuron, what will happen? More than two processes originate from a single axon. You can see, and multiple dendrites are also there. So multiple axon terminals and multiple dendrites from the cell body. So that is the demarcating factor of multipolar neuron. Multiple dendrites, multiple axon end terminals or synaptic end bulbs originating from the axon, originating from the cell body. So this is how the types of neurons are demarcated. Okay, is it clear now? Yes. Okay. So this is the type of four types of anaxonic, bipolar, unipolar, multipolar. Okay, directly from the neuron, no axon, two different cell processes, two different axons originating from the cyton, and then a small process originating from the cyton, emanating from cyton, and getting divided into two processes. That is unipolar, and the cell body is off to a one side. It is at one side of the cell process, and multipolar is what you have seen multiple times. In this case, what will happen is the cyton will give rise to multiple dendrites, and the axon will give rise to multiple axon end bulbs. Okay, good. So this is all about types of neurons structurally. Okay, always remember this is the structural classification. Okay, now functional classification, sensory and motor. Functional classification is sensory and motor. Functional classification, that was structural, now functional. Second is motor neuron. This is also known as afferent neuron. This is also known as efferent neuron. Sensory neuron will take the message or will conduct the message in the form of action potential or nerve impulse from the site. Suppose this is your hand to your brain. This is your brain. This is your brain. This is the hand or leg or effector or limb, you can call it. So this will be the direction of sensory. Means you are able to sense something, any temperature, any pressure, then heat. Everything is sensed by sensory neurons. Then the reaction to that, the movement of the hand or the leg or any body part, the movement is by motor neuron. Motor neuron, this is your sensory neuron. The movement is always due to the motor neuron. So if you have a motor neuron disease, what you will be having is simply paralysis. You cannot move your muscle, you cannot move your limb, right? So that is motor neuron diseases. Sensory neuron means you cannot feel something. If you are able to feel but cannot move, suppose you have touched a table with your hand, and you are able to feel that the table is extremely warm. But despite of knowing it, despite of feeling it, you are not able to take your hand off from the table. You are not able to take your hand off the table, you can say. So that means your motor neurons are not functioning properly. There is some kind of dysfunction in the motor neuron. That is the meaning of motor neuron, and that is the meaning of sensory neuron. So motor neuron diseases, like you must have heard of Stephen Hawking, right? So he was a great physicist, Stephen Hawking. So Stephen Hawking had a disease known as ALS. That was the disease which Stephen Hawking had. ALS stands for, just for your knowledge, I'm telling, amyotrophic lateral sclerosis. This disease leads to the degeneration of only motor neurons. Degeneration means death, cellular death of only motor neurons. And the life expectancy after the diagnosis of the disease is only 2 to 3 years. Means the prognosis of the disease is extremely poor. But it was a medical miracle that Stephen Hawking lived for more than 50 years, because he was diagnosed at the age of 23 to 24 and he lived more. I think he lived 75 or something like that. So he lived more than 50 years with the disease. Because what happens is, first of all, your motor movement will go off. In the end, the motor neuron of the, you can say, respiratory tract, means you wouldn't be able to deglute or swallow any food also, right? So, and you wouldn't be able to control your windpipe. You wouldn't be able to take breath because you are controlling the diaphragm, internal and what is called external intercostal muscles. So you would lose control. So thereby you cannot swallow anything. No swallowing of food. You cannot even breathe. So in the end, due to the what is called respiratory failure, respiratory failure in the end or conclusively leads to the demise of the person. But this was a medical miracle that Stephen Hawking lived for more than 50 years after the diagnosis of this disease. After diagnosis, he was the first ever patient to live with such a motor neuron disability. So death occurs due to respiratory failure. Okay. So that's the medical miracle. No doctor has been able to solve. I was I read a lot of research papers on this. But somehow, some way, okay, still it's a miracle. But the hypothesis was that his respiration continued, means the respiratory neurons, the neurons, the motor neurons which controls the diaphragm, the internal and external intercostal muscles, those motor neurons did not fail until the age of 75 when he died. So then it failed and then he died. So why didn't fail? What was the pathway? Still, it's a scientific concern, right? Area of scientific research, that what really happened, that all other motor neurons eventually degenerated, but that respiratory neuron, motor neuron that controls respiration and which is directly connected to the respiratory center in medulla oblongata and pons, they were not dysfunctional. They were quite functional throughout his life. Otherwise, there could have been nothing that could have saved him, right? Except the ventilator support. He could have been put on ventilator throughout his life, but that would have been of no use. So that was a medical mystery, right? So if you talk about neurology, there's a lot of things that people don't know. The maximum thing that people are unaware of is the thing known as consciousness. Consciousness. Can you tell me what consciousness is? Because I'll cite some examples for you. When you are sleeping, are you unconscious? When you are awake, when you are daydreaming, or simply dreaming? And what if you are in a medically induced coma or simple coma? How can you differentiate these four states based on the level of consciousness that you possess? So what is consciousness? It's very difficult to comprehend and decipher, you can say. The thing is, when we are awake, think from a very scientific perspective. Suppose I am awake, I'm teaching you, you are awake, you are listening to me. Okay? So the thing is, I can see, I can perceive things. That is known as consciousness, right? But when I am sleeping, getting, but that roles, when I am sleeping, how am I dreaming? How am I dreaming during the sleep? Means some of the neurocortical area in my brain is still active. We have the alpha, beta, theta, and gamma waves. So theta wave is mostly seen or observed in sleep cycle. Then there are two types of sleeps known as NREM, that is non-rapid eye movement, and then rapid eye movement. So when you are aware of your surroundings, fine. So during dream, I am also thinking something. And in my dream state, also you must have felt you are aware of your surroundings. Haven't you felt that? Yes. Because you will be running in your car or fighting with someone, so you are aware of your surroundings. So is it not consciousness? And when you are really given an anesthetic drug like ketamine, so what it does is it cuts off the synaptic supply, means synaptic connections. Directly it cuts off synaptic connections for some time and puts the brain in an unconscious state. But in that unconscious state, also it has been seen that the brain keeps on functioning. How can the brain generate alpha, beta, gamma wave? Via EEG. There is a technique known as electroencephalogram, just like electrocardiogram for the heart. Encephalon means brain, so EEG. So electrical activity is present in the brain. So the brain is kind of awake all the time. So the consciousness theory has not been proved decisively by any scientist till now, right? So I've read tons of research paper in consciousness. Still, there is a lot to be proven. One research came out 1 month ago in mice model, okay, because mice is a mammal closest to the human beings and very easy to replicate. They grow up easily, they reproduce easily, and easy to maintain. Quite you can say quite feasible there. So most of the research that I did was in mice model, and most of the scientists they do research in mice model only, right? In the beginnings, for suppose SARS-CoV-2 vaccine also, you will first test the vaccine in mice model, that is known as preclinical trial. Then you will be proceeding with preclinical trial. So you must have heard of clinical trials in the newspaper, right? Various vaccines are going on, are being recruited in clinical trial phase 1, phase 2 and all. You must have heard in the news, right? So Moderna's 1273 vaccine is in phase 2. Oxford and AstraZeneca's AZ-1222 vaccine is in phase 3 right now. Yesterday that Novavax vaccine got also in phase 1. Johnson& Johnson's vaccine has also entered phase 1 clinical trial. So, what are clinical trials or what is the role of mice? So, what happens is mice is a mammal, easy to maintain, easy to reproduce, and easy for us to know the phenotypes. And since it has got almost all the genes like us for the same types of proteins, so it is easy to replicate the results and think that it will be as safe in human as mice. So first of all, preclinical. Preclinical means not entering human. First you enter the mice model. In mice, we put the vaccine. Okay. And check for immunogenicity. Immunogenicity means is mice reproducing, not reproducing, producing enough neutralizing antibody. Because what does a vaccine do? It makes you produce neutralizing antibody. Okay. This is immunological concept. So if it is producing neutralizing antibodies via T helper cell 1 response and via memory B cell and memory T cell. Okay, so if you are not aware of this, just think of neutralizing antibodies as the antibodies which will bind to that specific virus or bacteria and will kill the virus. Okay, so antibodies are glycoproteins, proteins which are glycosylated or some Carbohydrate groups are attached to those proteins done in the Golgi apparatus of the cell, where the post-translational modification is done. After the protein synthesis process, which happens in RER, the protein is again modified in Golgi apparatus by adding glucose group, that is antibody. So if after a week neutralizing antibody is produced, then I can carry on the vaccine to the phase 1 trial. Now understand very clearly as a biology student, phase 1 trial assesses safety and little bit immunogenicity. Means now you have entered human model. Okay. Phase 1 means it is not preclinical, it is clinical trial. Clinical means you have entered human model. When you have entered human model, what happens is we will recruit first 10 to 20 critically ill patients. Okay. So in case of here, I'll recruit COVID-19 patients or high risk COVID-19 patients. So high risk COVID-19 patients like the workers who are working, the nurses, now they have no option. So they are very high risk patients that or high risk individuals you can say who can get COVID-19 at any point of time because you are a nurse and you are at a COVID-19 facility. So I'll administer that and wait for a few weeks and test whether neutralizing antibodies are being produced, number one. But number two is more important, more relevant. Whether it is safe, are they showing some toxicity, means are they showing extreme fever or heartbeat increase or kidney failure type or liver failure type? Is it safe for their body? So if it comes out to be very, very safe, then I'll go for phase 2 clinical trial. Okay. Then Moderna's vaccine, which is there, which you said you have heard, is in phase 2 clinical trial. Phase 2 will recruit some 200 to 800 individuals who are medium risk, and then it checks about immunogenicity, means how easily neutralizing antibodies are being produced after vaccine administration. If it is below a week, means before a week of vaccine administration, then you get the green tick to go on to the phase 3 clinical trial. Now phase 3 is the largest clinical trial, takes sometimes years to complete because you are supposed to recruit from 4,000 to 40,000 people, individuals, okay, who will be vaccinated, and then you will check both safety and immunogenicity, means formation of neutralizing antibody. Neutralizing antibody. You will check both. If it is fine, then you will submit your results to Food and Drug Administration USA, or if you are a European thing, then European Medicines Agency, which are the governing bodies for drugs, and then they may check all your data. If your data is all correct, they will grant you the approval, and then you can start commercializing your drug, means you can go for mass production of your vaccine and start selling it in the market. So that's how a vaccine is tested. Okay, or any drug, any medicine that you have taken has been tested for at least more than 10 years, you can say, because from chemical drug designing to testing a new drug, phase 2, first preclinical, in vitro in the what is called cell line, then you go for phase 1, phase 2, phase 3. It takes 5 to 6, sometimes 10 years for a specific medicine to come to the market. Everything has to follow a clear cut protocol. Okay, so that's why the neurological, what is called, research is also done in mice first. So in mice model, it was found last month that there is an area known as claustrum near the hippocampus region. There is a region hippocampus in the brain, hippocampus and olfactory bulb. Olfactory means ability to smell. These are the two regions which have stem cells in the brain and can divide. These cells can divide and form new neurons in human brain and mice brain. Other regions of the brain cannot have cell division. Those cells are either arrested in G0 phase, or if they have come to G1, right, S, G2, then M. Either they are arrested in G0 phase or G2 phase. It has been seen. So they cannot reach the mitotic M phase. Okay. So that's why when you have brain injury, it is kind of permanent because the neurons cannot divide. Same way the heart cells, the cardiomyocytes cannot divide. The liver cell hepatocytes can divide because they have scattered, again scattered stem cells. So that's the way, mystery that scientists are trying to solve, trying to take out the neurons from G2 arrest or G0 arrest and take them to M phase. So that's the research which is in its infancy right now, but it's going on in mice model. So there is an area in mice model which was speculated a few years ago called colostrum, means it's the area of integration of memory. It's an area of integration of memory, means various memories from various regions of the brain integrate here, and then we are able to recall things, and then we are able to recall stuff. So same area could be present in human beings also, but human brain research is still under ethical trial process because who will donate his brain, right? So after the brain is dead, something can be done. And mice brain, you can just kill the mice and get it done. So, but you cannot kill a human and start testing a brain, right? You have to wait for the death. So it's pretty slow, direct human brain. But still, mice brain, the cluster region has been found, which is really one of the epicenters of consciousness. So that region is the integrative center of all the, you can say, thinking or cognitive ability. So we all say that when we think, it's in the back of my head. That's a proverb. That means what? It is stored somewhere. It's not. I'm not able to recall. Because that time some neurons are able to communicate. So in the end, it depends upon the communication of the neuron. So the way you study, if you study with zeal, with passion, what happens is more and more synaptic connections are formed between the neurons or among the neurons. Then more the synaptic connections you will form, more the intelligence you will acquire. That's the case with Einstein, that's the case with Stephen Hawking. Because they had so much zeal, so much vigor to learn the subject that they entirely dedicated their life towards the very subject. And then they formed so many synapses that their brain became extremely intelligent. You cannot compare their intelligence to a common man. So that's why I always tell the students to love the subject. When you ask a lot of questions, then you open up your head, right? Do not only study for marks. If you love the subject, if your concepts are clear, marks will automatically come. Where will the marks go? But if you just try to mug up things and get the marks and then forget, you will never be able to grasp the subject. You will never have command over the subject. Your command will last only till the examination. After examination, if a person asks you something, you will not be able to answer. That's the only difference that it makes, right? You can say that is permanent knowledge and temporary knowledge, right? Topper but doesn't know anything after a few days of examination. So that's not the person anybody would want to be, right? Now motor neurons and what is called sensory neurons. So sensory neurons or afferent neurons, they form the afferent division of the peripheral nervous system. Now I had told you in the last class that the cell bodies of sensory neurons, they are located in the peripheral sensory ganglia. So ganglia means collection of cell body, ganglia plural or ganglion singular. That is the collection of cell body in the peripheral nervous system. And same way nuclei or nucleus is the collection of cell body of a neuron or of cyton I should say in the brain, CNS, right? So this is the term that you must be aware of. Now, sensory neurons are mostly unipolar neurons whose processes known as afferent fibers. Fiber means axon, okay? Extend between the sensory receptor and the CNS. So the human body has got almost 10 million sensory neurons which collect information from the external and internal environment. Okay, 10 million sensory neurons the human body has got. Sensory, which collect information internal and external. Internal means whatever problem you are feeling within. Sometimes it's pain that you are feeling, right? In the stomach, the pain in legs. So how that is perceived by your brain? That is internally you have what is called sensory neurons working, which are innervating your organs. That is also there. And then external one means which are present on my hand, my leg, right? Directly innervating the skin. So that is there, and internal means the pain that you feel in various parts. You can tell that, oh, this side of the body is paining. Internal somewhere that region is paining, the head is paining, the back is paining, leg is paining, ankle is paining. That is internal. Now collect organ from what is called information from internal and external stimuli. So the main region is what is called the main difference is there are two types of sensory neurons also. One is somatic sensory neuron, which monitors the outside world, and number two is visceral sensory neurons. Always remember, if you learn the things conceptually, it's very easy. Somatic sensory means hand, leg, and all outside world, and visceral. Visceral means always organs, deep rooted. Visceral means always deep seated or deep rooted. That is the word of words meaning in biology. So visceral sensory means whatever sensory neurons are innervating, whatever neurons are innervating your internal organs. That is known as visceral sensory neurons. Now, sensory receptors. What happens is there will be some receptors which are either the you can say processes or specialized sensory neurons or cells which monitor the sensory neurons. So we can you can say broadly categorize these receptors into three groups. One will be the interoceptor, which will monitor the digestive, respiratory, cardiovascular, urinary, you can say also reproductive, and provide sensations, what is called sensation of distension. Distension means stretch, how you are stretching, and deep pressure and pain. That is known as interoceptor neuron. So there are three types of neurons you can say based on interoception also. So based on the way you perceive different things. One is interoceptors. From the name you can say intero means internal. So this is visceral, right? Digestive, respiratory, reproductive, and internal pain, urinary, then internal pain, distension or stretch. So these are monitored by interoceptors. interoceptor neurons. So these are the special neurons known as interoceptor neurons. Number two is exteroceptor, means provide information about the external environment in the form of, you can say, temperature, touch, pressure, taste, smell, sight, then equilibrium, hearing. That will be your exteroceptor. Next would be, I'm writing down, exteroceptors, external environment. Extero means external, right? Now temperature, in touch, pressure. So these are the external things. Third is your very important proprioceptor. Very important. People miss this a lot. Proprioceptor. It's a new classification. It means that it monitors the position and movement of skeletal muscle and joints. Position and movement of skeletal muscle and joints. Very important. Movement and position. I'll explain this to you. Little bit different of skeletal muscles. Always remember skeletal muscles, not the smooth muscle which is under autonomic control, and joints. Now, what happens during the proprioception is those neurons try to understand they are always present in the joints which are felt by our body, which are which we feel, the elbow joint, the shoulder joint, the knee joint, right, the wrist joint. So suppose you are putting pressure on one joint, then that pressure is felt, and that pressure causes the body to adjust. Suppose your one knee has buckled, right? So automatically we'll feel that the other knee will try to take the entire weight or mass of the body in order to stabilize you. If your one ankle gets twisted, the other ankle or the other leg will try to stabilize you. That is known as proprioception, a very important sensory neuron capability that sends the brain the signal that this joint is not working fine. So the brain tries to send motor neuron signals, nerve impulses to the counter joint to balance the body. Your hand will try to balance you. Then your knee or your ankle will twist. That is known as proprioception, a very important way to balance ourselves and keep ourselves afloat in the environment, keep ourselves stable in the environment. So those are the things. Think how amazing your body is, right? It's quite amazing, our specialized nervous system. Scientists have only cracked, you can say, 20%, the best of the neuroscientists of the nervous system. Then you have motor neurons. Motor neurons on efferent neurons. They form the efferent division of the PNS. So the half a million motor neurons, what to do? They carry instructions from the CNS to the peripheral effectors in a peripheral tissue, organ or system. Now axons of these neurons that travel away from the CNS, that's why they are known as efferent, or axons which travel towards the CNS like the sensory, they are known as afferent neurons, right? Now the somatic nervous system includes all the somatic motor neurons. Again, you will be having somatic motor neurons, like you had somatic sensory neurons, motor neurons, which will monitor the external response to the stimuli. Means due to how do you, after touching, do you remove your hand or do you take your hand off? Do you run, or After feeling the pressure, do you shy away? Do you move away? So those kinds of things are all done by the somatic motor neurons. Visceral motor neurons are also there, which innervate all the peripheral effectors other than what is called skeletal muscles, known as visceral motor neurons. Innervates all the peripheral structures, means all the organs outside the CNS other than skeletal muscle, means which are not in our somatic control, which are in our autonomic control, autonomic nervous system. Means you cannot control your heartbeat, your kidney filtration, peristalsis. So those are conducted by the visceral. So how does the peristalsis happen? Again, the brain sends some signals, peristalsis will happen, but that is not under your control. How does your heart beat, high or low? Heart rate fast, heart rate slow, heart rate increasing, decreasing, all is controlled by the brain. So there are two nerves basically for the heart. One is the cardiac accelerator nerve that innervates the heart. It releases the neurotransmitter epinephrine. You might have heard epinephrine or adrenaline. So I'm giving an example. Cardiac accelerator nerve. This releases a neurotransmitter, which is also a hormone because it is released by the adrenal gland also. A very special case known as epinephrine and norepinephrine. These are also known as adrenaline and noradrenaline in the common tongue, right? So these are known as fight or flight hormones. They are made from the amino acid tyrosine, and they increase the heart rate, increase HR, the heart rate. So that's why they are known as, you can say, sympathetic nervous system also, that I'll teach you. Okay, sympathetic means which will excite you. So it increases the heart rate when you are running or when you need to run because somebody has pointed a gun at you or somebody is running behind you, or suppose your examination results are going to be declared tomorrow, you feel anxious, so then your heart rate increases, tension can increase heart rate. So all those things will cause the release of epinephrine from the cardiac accelerator nerve, which is again a motor nerve, again a motor neuron. Then next is coming to the vagus nerve. Now the heart rate has reached suppose 150 beats per minute. So is it good for the heart to stay that way when you are resting? No. Suppose you have returned from the games period back to the classroom. Now when you will be sitting for the first 2 minutes, you will be able to feel your heart throbbing, right? Pumping. That is known as palpitation, tachycardia. Now two things are there. Tachycardia. It's a specific medical term. Tachy means fast. You have the tachymeter in the car also to know the RPM of engine. So faster heart rate more than 100 beats per minute, tachycardia. Then you have bradycardia. Brady means slow. So heart rate less than 60 beats per minute. Or you can say 40 beats per minute is real bradycardia. Okay. So suppose you have tachycardia, which is known as palpitation in simple term, you are able to feel your heartbeat. But after a few minutes, means after 10 minutes, you won't be able to feel the heartbeat. It doesn't mean that your heart has stopped, right? You're just not able to feel your heartbeat, means your heartbeat has got reduced in number. The BPM has reduced and come down to normal 72 to 75 beats per second. Sorry, per minute. Now the HR, heart rate, has come down to 72 beats per minute. How did it happen? The vagus nerve, which also innervates the heart, releases a neurotransmitter known as acetylcholine, released from the synaptic end bulb of neuron. Now, this acetylcholine, also written as ACH, can reduce the heart rate. Thereby, your heart becomes normal in what is called the number of times it beats. That is how the motor neuron really controls everything. Can induce a lot of, you can say, peristaltic movement in the colon. Thereby, you need to go to washroom many times. Diarrhea can happen. Everything is done by this visceral motor neuron system. Then we can come to what is called interneurons. They are also very important. Interneurons. Again, this topic is sometimes left, right? But this is a very significant topic. Interneurons. In order to gain complete understanding of the nervous system. The name is inter. Inter means between the two. Again, biology is all about knowing English. That's all. If your English is good, there is no issues in biology. So interneurons are located between sensory and motor neurons. Always remember. Motor and sensory neurons. That is your interneuron. So there are 20 billion or so interneurons, also known as association neurons. These neurons are also known as association, and they are almost 20 billion in number, not 10 million and all. 20 billion interneurons. They are also known as association neurons. Okay. Now what happens is most of them are located within the brain and spinal cord, but there has been some interneurons which are found in the autonomic ganglia also, means the nervous system, the autonomic nervous system, which are located, the cell body of autonomic nerves, which are located in the peripheral nervous system. The main function of interneurons is integration, means they distribute sensory information and coordinate the motor neuron activity. The more complex the response to a given stimulus, then the more interneurons would be involved. Okay, that is how. If you are complex in the stimulus, then more interneurons are involved. If the thing is having pressure also, somebody has put a hammer also, and the hammer is hot also, so it is a kind of complex thing to understand for the brain. Then you will have more interneurons, so that more sensory neuron data can be transferred to motor neuron for the specific action. That is the role of interneurons. Okay. So any doubt till now? Types of neurons, sensory, motor, visceral, types, interneuron. Any query? Okay, good. So now you must be feeling quite interested in neurology, right? So much is there to explore in neuroscience that people are still unaware of, and if you make sure, or if you think logically about your body, you can think of these processes happening. Right? So that's what biology is all about, knowing about your own system. So next up we will start neuroglia. Now people talk about neurons a lot, but neuroglia are that much important too. Neuroglia. So they are 3 to 4 times more in number than the neurons. Means I told you there are 86 billion neurons, so you can just multiply 86 billion by 3, okay, or 4. So that is the number of times they are present. But neuroglia cannot conduct nerve impulse. They are non-excitable cells. Okay, no nerve impulse. They cannot conduct nerve impulse. Now, what is their use? Their use comes in the nourishment and maintenance of neurons. In simple terms, maintenance of neurons. That is where you can say the type of neuroglia and all the neuroglia, you can say concomitantly, they perpetuate a common function, that is nourishment and maintenance of neurons. Now there are two types of neuroglia. One that is present in, you can say, CNS, and one that is present in the PNS. So I'll just write it down. So neuroglia, CNS, PNS. Now under CNS you have number one astrocyte, number two is oligodendrocyte, number three is, you can say, ependymal cell, ependymal cells, number four is, you can say, glial cells or microglia. So these are the four types of neuroglia or neuroglial cells present in the CNS, that is brain and spinal cord and spinal cord. Under PNS you have two types, Swan cell, then satellite cell. So these are the two cells or two cell types present in the peripheral nervous system, which are, they have a common function of nourishing the neuron in the peripheral nervous system. And these four types of cells nourish neuron in their own way. They have different functions of during the nourishment, but they all nourish the neuron one way or the other in the central nervous system. Okay, so this is neuroglia. Now it will become more interesting. Now coming to the, you can say, astrocyte. Astro means star shaped, astrocyte. Now there are, you can say, if you want to divide even further, you can divide it into two types, right? Astrocytes. One is fibrous astrocyte, one is, you can say, protoplasmic astrocyte. Now fibrous astrocyte is present in the white matter and protoplasmic is present in the gray matter. Now what is white matter, what is gray matter in nervous system? Again a very easy question, but comes along and sometimes people get perplexed, but there is no reason to worry. White matter means wherever you have a lot of axons. So axons are coated by myelin sheath, and myelin sheath is made up of mostly lipids and proteins and highly lipid in nature. So the lipid is made up of three fats and glycerol, so it is mostly fat only. So lipids, they are perceived or they look whitish under the microscope. So wherever you see a lot of white appearance of the neuron under the microscope means the lot of axons are present there. That is the meaning of white matter. Whereas if you have a lot of cell body, cyton, dendrite, axon, end bulb, okay, which is also known as telodendria, means which are unmyelinated. Unmyelinated. So that means that due to unmyelination, there is no fat layer on top of these specific structures and they appear almost gray in color under the microscope. So wherever you see grayish color, when you are analyzing a set of neurons, you will know that these and these may be the cyton or dendrite or the telodendria. They do not have any myelination. That is the meaning of gray matter. Okay. Sometimes what happens is the myelination is not present on axon. There are some neurons which are known as type C fiber. Fiber means axon. So type C fiber containing axons are unmyelinated. Okay. There are three types: type A, type B, and type C. That I'll teach you in the next class and further. So type C fiber axons are unmyelinated in nature. So these axons will also appear gray. So white matter means wherever the axons are myelinated. That is the direct rule of knowing white matter. Okay, that is how you understand what is white matter. So any doubt till now? White matter, gray matter, function of neuroglia, 3 to 4 times they are present more than the neurons, number of neurons. Okay, good. So in the next class, we will cover the types of neuroglia and the current generation, that is known as the nerve impulse generation, the depolarization, the repolarization, and the hyperpolarization in case of neurons. That how is potential difference generated? Because in order to generate current in physics, you might have read that you need potential difference. Now, how is potential difference generated in a neuron? And what are the various functions of this oligodendrocyte, astrocyte, ependymal cell, Schwann cell, and you can say satellite cell and microglia in PNS and CNS. So we will discuss all this in the next class. If you have any doubts when you will be reading your book or from net, you can always put your doubts in the group directly to me and I'll just reply to you as soon as possible. Okay. Okay, so we'll meet in the next class. Thanks a lot.